Hey everyone, and welcome to The Explainer. Today, we're heading out on a virtual nature walk. If you just glance outside, nature can sometimes seem completely chaotic, right? Just a dizzying array of shapes, sizes, colors, and sounds. But what if I told you there's actually a hidden, highly elegant order to the whole shebang? In this explainer, we're going to decode the living world, transforming raw, everyday observations into clear scientific categories. So grab your virtual notebooks because we're stepping outside to uncover the rules that govern the plants right in our own backyards. Here's our trail map for today's inquiry. We'll start by observing the living world, then move into grouping by common traits, unlocking leaf venation, going underground to check out plant roots, and finally, piecing together the leaf-root connection. Let's jump right in. All right, kicking things off with Section 1, Observing the Living World, where we'll cover Activities 2.1 and 2.2. We're placing ourselves right alongside Dr. Raku and Maniram Chacha as they guide these students out in the field. Notice how completely engaged the students are, getting up close and personal with stems, leaves, and flowers. But, and this is absolutely crucial, they are doing it without disturbing them. They're paying attention to the weather, they're carefully listening to unique bird calls, and they're realizing that every single organism out there contributes to the massive diversity of the living world. This right here is active observation in practice. And taking those observations builds our very first data set. We've completely filled Table 2.1 with our initial plant observations from Activity 2.1, and it is super methodical. For common grass, we documented a soft, thin stem with single, alternate green leaves. Contrast that with the Tulsi plant. That one has a hard but thin stem, leaves pointing in completely opposite directions, and pinkish-purple flowers. Then you've got the hibiscus with its hard stem, broad leaves, and vibrant red flowers. And finally, the massive neem tree, featuring a hard and thick stem, smooth leaves, and small white flowers. We're literally taking the wildness of nature and systematically recording its distinct features. Moving smoothly into Section 2, Grouping by Common Traits, tackling Activities 2.3 and 2.4. So for Activity 2.3, the students are actually pooling their data cards together on a blackboard. When you bring multiple observations together like this, you can start asking the really big questions, like how do we categorize all of these? Do we group them by how tall the plant is? Or maybe by whether or not they have flowers? Grouping things together is basically the foundational bedrock of biology. It lets us see these overarching patterns that are totally invisible when you're just looking at a single plant in isolation. Which brings us to the crux of Activity 2.4, categorizing our observations into highly functional groups based simply on their stem structure and growth habits. So herbs are our small plants with soft green stems, like a tomato plant or that grass we observed. Then we have climbers, which have weak stems and literally need some sort of external support structure to grow upwards towards the light. And finally, creepers, which also have really weak stems but choose to spread and creep horizontally along the ground. Boom! Just by checking out the stem, we've organized a massive chunk of the plant kingdom. Next up is Section 3, Unlocking Leaf Venation, diving into Activity 2.5. Now, if we zoom in on the leaves we just collected, you're going to notice these tiny, intricate lines running inside them. Think of them like microscopic highways. These are veins, and their job is absolutely critical. They carry water and food to every single microscopic part of the plant. But if you look closely at different leaves, you'll quickly realize these highways aren't all built the same way. This specific arrangement of veins on a leaf is what scientists call venation. Comparing the two master designs of nature side by side brilliantly reveals our findings for Activity 2.5. First, we have parallel venation. Here, the veins are arranged completely parallel to one another, running in straight lines right along the leaf. Think of a blade of grass or a wheat leaf. Then we have reticulate venation. In this design, the veins spread out extensively like a complex network, or a net-like pattern, exactly like what we saw in the hibiscus or neem leaves. It's a beautiful structural difference that, once you know it, you can spot in seconds. All right, let's get our hands dirty in Section 4, going underground to look at plant roots, which is Activity 2.6. Okay, let's dive into this underground comparison. When we wash away the dirt, we find two primary root systems. First up is the taproot. This is a thick, main root growing straight down from the base of the stem, with smaller secondary roots branching off it. Turnip, carrot, and mustard plants, they all have powerful taproots. On the flip side, we have the fibrous root system. There's no central main root here. Instead, it's a big, bushy bunch of thin roots all sprouting out from the stem base together. Onion, wheat, and our common grass rely heavily on these expansive fibrous roots. Which brings us to the final piece of the puzzle, Section 5, the leaf-root connection, wrapping up with Activity 2.7. So here is the absolute most important question of this whole explainer. Does the pattern on a leaf tell us what's hiding underground? Think about it for a second. Can we actually predict the invisible root structure of a plant simply by looking at the visible network of veins on its leaves without ever picking up a shovel? Let's check our final data set to find out. And here is where it all comes together. When we look at our completely filled Table 2.4 data, it reveals an absolutely stunning biological rule. Take the mustard, carrot, and hibiscus plants. We observed earlier that all three of these have reticulate, net-like leaf venation, right? Well, guess what? Every single one of them has a taproot. Now look at the grass and wheat. They both have parallel leaf venation, and they both feature bushy fibrous roots. The data is incredibly clear here. Reticulate veins literally always pair with taproots, and parallel veins always pair with fibrous roots. You can basically read a plant's underground secrets right on its leaves. How cool is that? So, now that you know the secret language of leaves and roots, what hidden patterns are you going to discover in your own backyard? Science isn't just something trapped in a textbook. It's waiting for you right outside, ready to be observed and categorized. I highly encourage you to step outside, take a really close look at the next leaf you see, and know with total certainty what kind of roots are holding it down in the earth. Thanks so much for joining this explainer, and keep on observing.